Good afternoon, everyone. I hope all are learning something new during the COVID time. My name is Albin Jose, and today I am here to speak about application of SAS in clinical trials and answer some of the questions which might rise in your mind during the presentation. First, I will explain in short about the basics of SAS, and then I will take you through clinical trials and the data extracted from it. Later, I will show you the roles of SAS in clinical trial with few programs. Now, to help everyone build your career, we will also see some facts and figures. And also, I will be speaking what you will learn about the clinical SAS from Sankhya. Now, speaking about SAS, in other words, it is statistical analysis system, which means it helps in accessing the data, managing the data, analyzing the data, and presenting the data. It is a tool in which we can access any format of data that can be Excel sheet or a text file. Then we can clean the data and validate it, or I can say I can manage the data. We'll do some analysis to identify what the data wants to tell us, and in the end, I will create a report. Moving forward to understand about the clinical trial, clinical trial does not work just by any doctors prescribing the pill to a patient. We treat patients as a human being, not as a test subjects. There are different participants in the trials. They come, they participate with their necessity and their consent. Here, this picture you can see the doctor is writing something on the paper. He is actually collecting the data. So, clinical trials are planned experiment which involves patient and it helps in identifying the proper medications which helps the future patients to cure disease without any complications or side effects. Let's dig in more about the process of clinical trial. The process of clinical trial begins with the approval of protocol, which contains all steps or plan to conduct a clinical trial. The protocol includes everything about the clinical trial. We then select the medical investigators and get the process approved. Without patient, clinical trial is nothing. We recruit patients and inject the medicines in which we were testing about the drugs. We collect the data, enter it into the database, and validate it. We analyze the data with the help of statistics and create the reports for publication. In the end, we file the reports and register them. Now let's understand about the phases of clinical trials. There are four phases of clinical trials. The first phase will take about months to 1 year. Now this depends on this type of study which we are collecting the data for. We'll be recruiting healthy patients, about 10 to 20. It can go up to 50 also. We check whether there are any side effects or not. We'll be assuring the safety and efficacy of the drug. About 70% moves to the next phase once we get an approval from the regulatory authorities. For phase 2, it takes about 1 to 2 years of duration. And here we recruit about 50 to 500 diseased patients. In phase 2, we will monitor the safety and efficacy. However, we will also find the most effective drug dosage which helps the patient to cure without any side effects. From phase 2, only 33% of drugs move to the next phase. And always remember, after each phase, the reports are sent to the regulatory authorities and they will be approving to move forward to the next phase. Now, the next phase is phase 3. It takes about 3 to 5 years to complete. In this phase, we target for the larger number of groups of diseased patients. We will check the safety and efficacy, but also we check whether there is any risk in the larger group. The answer for what risk. Now it is whether the patients have prehistorical disease like blood pressure or the diabetes. Now it can give a different effect because the drug which they are taking for prehistorical disease with the drug inducing during the trial. That is, there can be any drug to drug interaction or drug to body interaction. This will be checked in the phase 3. Once the phase 3 is completed, the application is submitted to the regulatory bodies to get an approval for the marketing, the drug, and make it available to the public. We think that the work is done here. No, we still have another phase. Remember, I said four phases. So phase 4 is also known as post-market surveillance. In phase 4, there's no fixed duration. As long as the drug is in the market, the phase will continue. You might have heard about some drugs being banned in the market, like Vicks Action 500 and the recent one, Rantac. This happens because of the change in the atmosphere and the genetical change in the different generation. That is why the drug can have a different effect. Here, they focus on safety and efficacy. They also check whether there are any rare side effects or not. I have been saying about the regulatory bodies again and again. Now, who are these regulatory authorities? These are the regulatory authorities who take control of the drug which are being released in the market. Now, the regulatory authorities came into the picture because the incident happened due to the thalidomide during the Germany. This drug was released for the pregnant ladies, labeling it as safe for the pregnant lady and child. It was to cure the mother's sleeplessness and morning sickness. But after the after effects had a big regret as the child was born with pachymelia. That is, the kids were born with deformity. They were born either with any deformed, without any limbs or a deformed body. Now, to avoid these kind of drug release, these regulatory authorities take a strict action. Here are some of the regulatory authorities which we have, like DCGI from India, we have FDA from US, EMEA from Europe, MHW from Japan, and we also have MHRA from UK. Now let's see what the clinical trial data contains. The clinical trial data involves preclinical data or animal data. That is, the data are collected while the drug is injected into the animals. Then we have structural activity data, which includes the molecular or the compound structure information about the drug. It also includes laboratory data, where we conduct different tests like urine test, blood test, stool examination, red culture, and various other cultures too. The result of these tests are collected as data. We have literature data, the data where we collect through word of mouth. Now, the medical investigators will be asking different questions like, did you have any pain in stomach or headache after taking pills? Did you have the medicine or did the medicine made you sleepy? Now, this helps to identify the side effects which might arise from the drug. And the last one, that is the clinical data which we collected while the patient is available. That is the patient's the data like heart rate, we have blood pressure, we have ECG, and so on. Now these data are collected and stored in data warehouse. From the data warehouse, the SAS programmers will clean and validate the data. They will do required analysis and create reports. From these reports, the regulatory bodies make the decisions, or I can say they make the data-driven decisions. Now, let me show you some programs, what we do using the SAS. Now, in this interface, you can see the SAS Studio Interface. Now, here I am going to explain you first about the data which we are going to use, that is the sashelp.heart. Now you can see here I have the data named as sashelp.heart. Now sashelp is the library, or you can say a folder in which all the data are being stored. So you can see the left hand side where I am scrolling. This is the sashelp library, and these are the different data which have been stored in sashelp. Now, this is not only the alone the SAS Studio interface which we have. We have different other interfaces too. Like we have SAS Enterprise Guide, we have SAS Mainframe, we have the newly launched SAS VIA. Now, these different interfaces help us to do the data analysis. Now, let's dig in more about the data. That is, you can see somewhere around more than 5,000 rows and 17 columns in it. Now, what are the different 17 columns? I have status, I have net cost, I have sex, I have height, I have weight, I have PP status, I have weight status, and so on. Now, these are the different 17 columns. Now, let's go into the programming. Now this is one program which has only 3 lines. Now what does this program do? This program copies whatever was there in the SAS.hard to a new data or new table named as 1. Now you can see here the codes which I have used. That is, I have data 1 set SAS.hard and then run. Now always remember, in SAS there are 2 steps. That is, one, I have a data set which I am showing you right now, and the other which I have is a proc step. I will say about the proc step later. Now, what does it do in data step? Now, the data keyword will help you to create a new table named as 1. The set keyword will help you to read from the pre-existing data. That is, I have a hard table from the SAS library, and then I will end it with run. So all the steps always either end with run or quit. Always remember that. So here I am beginning with a data keyword and ending with a run. So it is a data step. Now let's execute this program and see what happens. I can see that it has been creating a new data set with 1 and the same result. You can see that I have the same number of rows and same number of columns, and all the values and everything will be copied into 1. Now there is one more thing. You can see that I have different tabs here. I have code. Code is where I will be entering the data or I will be writing down the programs. Then I have log, where I can see 3 things. That is, I have errors, warnings, or notes. Now errors will give you any warning, or it will be shown in red color here. So that is, if I'm going to make any mistakes in code, I will be able to check it in log with errors or warning. Then I have notes where it gives the brief description, what has been done. So here it shows that the dataset work.1 has 5,209 observations with 17 variables, and also the rest how much time it has to for processing. Now let's understand about the proc step. Now here I have one of the proc step which I am going to speak about is the print proc print. That is, it is to show it in the report format. Now there are different other proc steps where I will be taking you through different some of the proc steps today. So here I have just used proc print data is equal to saxl.r, and here I am using where status is equal to death and age at start is greater than 15. So I am taking only a portion of data from that. That is, I just want where the status is death and age at start is less than or 15. Now these are the variables which I want. I don't want all the 17 columns from the saxl.hard. I just want these 5 columns from them. So let's execute this program and see what is it I get in the result. I get it in the result with these things. That is, I'll get it into the report format with whatever I require and all the values within them. So I have all the how many values will be there. Now how can I check it? Now here, if you remember, I have used only portion where I require only portion of the table. So I can check it and log how many observations have been written. These are the number of observations which are read from the SAS and podcast. So I have only 941 observations in this report. Now you understood about the proc report. Now the proc print. Now it's not only proc print I can use. I can use different other programs. Like I can use it for statistics. Now I say for simple example, I want to find out the average depth of age of the patients. So I can use proc means there. So when I execute this program, what is the result I get? I get only with different, that is, I have segregated classified into sex for males and females, and this is the mean which I get, that is 71.56, that is the mean of age at death. I have a column named age at death, that is this one, and you can see it in here with these columns. So this is the mean report which I get. Now for female I have 71.56 and for male I have 69.69. Now I also get different other statistics, that is I can get standard deviation. I also get minimum value, that is minimum value For age of death in the classification of female, I have 36. For male, I have 36. For maximum value, I have 96 in female and I have 91 in male. That means female tend to live for longer period. Yeah. So let's get into the different other programs. That is, I can not only go with the average, but I can go also with the number of count. So I want to know how many times unique values are being occurred. Now you can see in the first column here, I have status, I have death, and I have alive. Now I want to know how many dead patients are there, and I want to know how many alive patients are there. So I can use pro free. Now this is the program which I'm going to use. Now let's execute this program and see how many are dead and how many are alive. I have 3,218 alive and I have 1,991 dead. Now this is not only the result which I can find out, I can find out more. Now say I want to see how many different kind of death cause are there and what are their number of occurrences. So I can use this program. Let's check it out. Now you have been seeing that I have been using semicolons in all the end of the statements. Now those, whenever I am starting with a keyword and ending with the statement, I can see that those are known as statements. Now here you can see the cause of death. Now cause of death, that is the death cause which I had used. There are 5 different unique values to it. I have cancer, I have cerebral vascular disease, I have coronary heart disease, I have other, I have unknown. And I have found out how many times it is occurring in that specific column and what are the percentage of this. Now I have some missing values also. How many are there? There are 3,218. Now where did you saw that? Those are the alive patients. So that's why it is missing there. Now, other than these proc-freq and proc-means, that is the statistical programs which statistics which we use, we can also use different graphs. Now here I am going to show you the different scatter plot which I have used here. Now you can see in here the data which is about showing about the number of deaths per the age factor. I can see the age in the y column, y axis, and the number of deaths. So you can see here the dots, various dots. I can see it in this graph too. Not only with scatter plot, I can use the bar graphs too. Now this programs help you to see the bar graphs. Now there are different types of bar graphs. Now here if you see, I have classified with sex, that is I have male and female, and I have also took the age at start, which is from 50 to 60. So this is the number of age which I am looking into, and for that I have the frequency, cumulative frequency, percentage also, and the cumulative percentage also. This way also I can represent it in the report. Now not only with graphs, that is age of the graphs, I can also look into with the pie charts. Now in this program, I am using the pie chart where it shows different male and female candidates. So I can see here in the pie chart, there is the values, it is starting from 40 and it is ending till 45 and the percentage. So blue one is the male candidates and pink one is the female candidates. Now I will show you something interesting that is extra. I can create maps with it also. That is the maps and it is showing the populations of Asia. Now this is something interesting, right? How can anyone show with a report? That is, I have only somewhere around 9 lines of code and I am able to create a whole map of Asia wherein I can see the different graphs in there who are more with the population. Now this is the population of 2005. Now this is one example which I wanted to put you out with. Now you understood about how the programs are being conducted and also how the programs can be written down. And also you might have understood about the clinical SAS. Now let's check the market of the life science and the pharmaceutical industry. Now I spoke about data which we use in clinical trials. We use analytics here. There are tons of data being created as there are 3,39,863 research studies in 214 countries, which you all can update it from clinicaltrials.gov website. And as industries will be upgrading themselves with the technology due to the recent epidemic, the data will be more accurate and it will increase in numbers too. Now, the life science market is segmented into four different segments. That is, we have pharmaceutical biotechnological companies, we have medical device companies, we have research centers, and we have third party administrators. The pharmaceutical and biotechnological companies segment commanded the largest share of the market in 2019. Now, this can become a cause to high utilization of analytic solutions by the pharmaceutical and the biotechnological companies. And we see in the graph that next years it is going to increase. It is growing from all the regions. That is, we have North America in blue, we have Europe in green, we have Asia in parrot green, and we have rest of the world in yellow. So it is going to increase in a dominant manner. Now you can see even with the different numbers. So the life science market is to be projected to reach to 33.2 billion USD by 2024. Now the market seems good, right? But then what about the salary? At least by the end of the month, I should get a greater number of digits in the bank balance, right? I researched and found in pay scale that as the experience increases, the pay also increases. When I searched in different job portals, I could see that SAS programmers of more than 10 to 15 years of experience get a pay of more than 15 lakhs per annum. Now, we can see that the average salary by the popular employers in India too, on the right hand side, right? Now, the highest value here shown is 7 lakh 37,000, and we have at the least which shows 3 lakh 25,000. Now, these figures are as per the experience. Now, what would I like everyone to do is to go and search in different job portals and check out the salary packages you can get for clinical SAS programmer or a SAS programmer or a statistical SAS programmer. Now, till here you have understood about this. Now, let's understand what you will learn of clinical SAS in Sankhya, from Sankhya. We have different courses available. Before learning clinical SAS, we should be good with base and advanced because we will be using these programs in clinical SAS. We'll be learning about CDISC standards. We'll learn about different domains in SCDM. Now, SCDM is Study Data Tabulation Model. We'll be writing the codes and creating different domains like demographic domain, which contains the details of the patient. We have trial visits domain, which contains the details when the patients have visited the site. Exposure domain, which contains the details of what kind of drug was being prescribed during each visit. We have laboratory domain, which contains the details of the test and research, the results of the laboratory test. We have vital signs domain, which contains the details of blood pressure or heart rate and etc. And we have trial domains, which contains the planned treatments for our patients. Now, once we have completed the STTM, we'll move on to ADAM. Here, we'll learn about how we are going to convert this STDM domains to ADAM domains. Now, these are the domains which we are going to create, are the different tables which we are going to create with the help of SAS. Whatever the data which we have achieved, we will be creating those STDM domains. And from the STDM domains, we will be creating different ADAM domains. That is, we have ADSL, we have ADAE, we have ADLB, and so on. It is based upon the requirement which we want to do the analysis. ADAM is analysis data model. Now, what is the requirement? What is the requirement to convert these domains or ADAMs? We'll be learning those all things also in the training. Now, we also learn about the tables, listing, and figures. That is, the different tables, listing, and figures which we'll be creating with the help of SAS. We'll see some figures being created. We have seen the different procedure steps which I have shown you before. So we will be learning those kind of steps. We'll also learn different other similar graphs which will be help in creating the reports. Now, I hope I have answered some of your questions or most of your questions which comes into your mind, like what is clinical SAS, what is SAS, about the programs, whether it is hard or easy. Now, the next question which I will ask is by Sankyana. We here are a SAS accredited trainer. We provide classroom training and live web training. We also focus on candidates not only to pass the certificate but also to be an industry ready. We make the students go through different tests provided by us. We have 11 online question exams. We have also which will be helping different exams, that is it will be able to help them to crack the certifications. Not only that, we also have trainers who will be helping you out with the mock interviews too. Now the decision lies upon you to make a move and build a career. Now every journey has an end, and here I am to end the presentation by thanking everyone for spending your time listening to me. And I hope everyone spends the time wisely for developing yourself and looking out something prospective during and after the COVID time. Let's spend the time planning the future and also making it bright. So thank you everyone and have a great rest of the day.